So first of all, I'd just like to thank the organizers for inviting me to talk. And I'd like to thank Paula for introducing the CRISPR-Cas9 system, which makes my work a little bit easier here. And I want to also say that really this is a collaboration between a number of different labs. As Rowena discussed, it takes a village to do these kind of studies. So we have a lab at Temple, but also we're working with the Tulane Primate Center and with Case Western University. So I'm going to start with my community summary. So what is the key research question we're going to ask here? So really, can proviral DNA be edited in a primate model of HIV using CRISPR technology? And we're using our technology is an in vivo IV inoculation directly into animals. So the CRISPR technology can be widely distributed in various tissues by viral delivery system, and it can remove fragments of viral DNA from cells and tissues. Hopefully I'll convince you of that by the end of my talk. So why is this important? As I said, it's really the first time in a large animal model that we've shown an IV inoculation of CRISPR that can edit a target gene, and in this case, we're targeting and trying to remove viral DNA. And how is this related to cure? So of course, we all know HIV DNA integrates into the host genome, so we can use this genetic strategy to lead to a potential cure of HIV disease. And really, we're excited about this because we think it's the first step towards a permanent elimination of virus, and it really gives us insights into future clinical studies. And I would say that, this is a great meeting because I'm hearing a lot about, host genome and host editing and also viral genome. So I think, the combination of these is going to be important. Okay, so how is our system, what are we doing in our system? So like I said, we're using the CRISPR-Cas9 system, and we're targeting two portions of the viral genome. We have gRNAs that are specific for LTR and for GAG. So in this way, we can remove large fragments of the viral DNA from 5' to GAG, 5' LTR to 3' LTR, GAG to 3' LTR. So we're trying to remove big sections. And in this way, as we heard from Paula, we're cutting in two sections, removing a large portion, and then by non-homologous end rejoining, we get a large deletion. And we think in this way, we get less probability of escape. And so we designed these gRNAs using rigorous bioinformatic technology to look for conserved regions that are highly conserved, but also looking to make sure that there's not going to be any off-target effects in the host. So our system is an all-in-one delivery system, and delivery is a big issue, and I'm really excited to hear everyone's opinions on these tomorrow. We are using the AAV9 system. So in this way, we're delivering Cas9 and our two gRNAs into cells simultaneously, and this just shows you a transfection that we can see the Cas9, we can see our two LTR GAGs present in cells. So our system, is producing the products we want. So then we wanted to look at transfection and transduction, like I said, by AAV9. This is just in a cell line. I'm going to get into the bulk of this data. This is the same system we used in the paper that you had discussed in the mice model. I'm not talking about any of that data, but if anyone would like to discuss that, I would be happy to discuss that as well. So what we're really seeing, what we're seeing is we can deliver by AAV9 and we can truncate the virus. So like I said before, there's 3 different portions that we're looking at once you remove the virus. So what we're doing is, in here we're showing 5' to GAG, and also we can look at GAG to 5' LTR. So basically it's after the section is removed, we use PCR amplification to amplify the piece that's left over. And this also has a PAM sequence, so we know exactly where we have edited. So it's kind of I call it like a marker of editing. And all of the data I will show you has been PCR amplified and sequenced, because we want to make sure that we have the breakpoint and we are showing the data, so everything is verified. So getting into the kind of the crux of my study is that we used a non-human primate model. In this study, we used in Chinese rhesus macaques infected with SIV MAC239. We had an N of 8. We used daily RNA administration. As you can see at the top, we used the frontline first regimen. We then did an ex vivo gene editing to confirm that we can edit PBMCs prior to in vivo administration. And our in vivo editing is a one-time injection. In this system, it's 10 to the 13th GC per kg. And we did this in a pilot study of 3 animals, and then followed up after 3 weeks with a necropsy. We looked at biodistribution, safety measures, histology, and other measures of excision, and I will show you some IPDA and EDITS, which is RNA and proviral DNA. So this is really the ex vivo DNA excision. So what we did is we took out PBMCs after the animals were infected. We gave them AAV9 with our vector just to see if we could excise the viral DNA. And you see here, this shows you the full-length LTR. This shows you the truncated 5' to GAG. This is our expected product. So in this case, it's untransduced and transduced. So you can see the gene product here. And this is the other way, GAG to 3'. So this is what we're amplifying after we remove that section, as well as showing that we are getting Cas9 and LTR, the gRNA expression of the LTR and GAG gRNAs in these ex vivo PBMCs. And again, like I said, everything here is sequenced to confirm that we have proper excision. So this is really the crux of the matter here. This is the in vivo gene editing. We did this in 2 kind of pilot studies. We did an N of 2 that had the in vivo CRISPR with 1 control, and then this side over here shows a 3rd animal with 1 control. So to start over here on the left, we have 2 animals that are positive. Again, same as the ex vivo. We are looking at the truncated GAG, LTR to GAG products here, and the ones that had in vivo excision, no expression or no cleavage, of course, in the control animal that did not receive the CRISPR-Cas9. And we see the other way, the GAG to 3' LTR. We showed the expression of the Cas9 in both of the gRNAs. And this is in blood from the animals. Again, we showed the same thing with the other animal, KB88. So of course, one of the major questions is we're taking the AAV9 and inoculating it in vivo, but what is the distribution in tissues? So we've looked at the Cas9 DNA in tissues, and this is by digital droplet quantitative PCR of the 3 animals. And I will say, of course, the control animal did not have any Cas9 DNA present. And you can see that it's widely distributed in all tissues we've examined, with varying levels. But we see it in bone marrow, spleen, gut, lymph node, thymus, liver. We see it in brain, kidney, heart, testes, spinal cord. So we're getting, distribution in multiple tissues, lymph node and including lymph node tissues. So then we really wanted to know, so this is grinding up tissue and doing a qPCR or digital droplet PCR. We wanted to look at DNAScope to see in those tissues where is the Cas9 distributed. So this is from 2 of the animals, mesenteric lymph node and spleen. And in this red color, here's the distribution of the Cas9 DNA. And we quantified this in the different animals. So we are seeing, targeted Cas9 expression and delivery. So I think this is one of the more important slides because we saw it in blood with excision, but what about tissues? So this shows you some of the lymph node tissues. We're looking at spleen and lymph nodes, and you can see here at the top, this is the product, the truncated LTR to GAG. And we're seeing that in the animals in the spleen. We have 2 different pieces of spleen. And these have all been repeated at least 2 times to confirm and sequence for confirmation that this is the correct product. And at the bottom here, you can see in lymph nodes, we have some lymph nodes that have excision. There's some that we did not detect excision, but this could be a sampling error. We're going back and, lymph nodes are tiny and fatty. This could be that we just didn't get the right portion, so we're repeating that now. But this is very, very promising data showing lymph node excision. So this slide is very busy, but this is, I think, one of the most important slides that I'm going to show you is this is really showing for the first time viral excision in vivo tissues. And we're showing here on the left, this is lung. So we show excision in LTR to GAG product and also GAG to 3'. And again, nothing in the animal that did not receive the CRISPR-Cas9. Over here, we see cervical DRGs, and we see spinal cord and testes. I know I got this at a question at Croy. Does it also cleave in the testes? And I can now report that yes, it does, and in the spinal cord. We also see excision over here in the gut tissues, although variable, but we can report that. And I think this to me is very important. And we do see viral excision in the brain. And this was pretty consistent. Every brain region that we've examined so far in the animals show viral excision in both the LTR to GAG and the GAG to 3' LTR. So of course, everyone wants to know, what does this mean as far as, viral replication? So this study, I will say, I want to make clear that we did not remove antiretroviral therapy. The animals were on antiretroviral therapy the entire study. This is just proof of concept that we can inject AAV9, deliver CRISPR-Cas9, and edit viral DNA in blood and lymph node and deep tissues. So this just gives you a little flavor of what we're trying to do as far as viral reservoir, because I think I'm really excited to hear our keynote speaker later and see what he has to say about this. We used the new intact proviral DNA assay developed in his lab. And so what we showed, this is lymph nodes. So we did lymph nodes prior to treatment and after treatment. And we looked at the intact viral DNA. And we see that we have various degrees of decrease of the viral DNA, intact proviral DNA. We have a decrease of 38%, 77%, and 95% in the animals that receive CRISPR-Cas9. I will note we do see a little bit of a decrease in the animal that did not receive CRISPR-Cas9, but I'm assuming this is viral decay associated with ART. We also worked with John Carn's group at Case Western University, and we were doing his EDITS assay, and his EDITS assay is really looking at SIV envelope messenger RNA in memory CD4 T cells. And I will state that this is a single time point at necropsy, so I wanted to present this data, though, because I think it parallels what we're seeing in our animals. So KK09 is the animal that did not receive CRISPR-Cas9. So you can see after concavalin A stimulation of the cells, the amount of memory CD4 positive cells, so the RNA induction, and you can see there's about a 50% decrease in the RNA induction in the KM77 and KP43, which are 2 of the animals that received the in vivo CRISPR-Cas9. We also did a viral recovery assay, and I will say that this is not a QVOA assay. This is really an assay where we took PBMCs at necropsy. We put CEM cells in there for co-culture, and we just kept the cells alive and see if we could get any virus to grow. We didn't do any latency reactivation. We didn't do any of that stuff. This is just rudimentary viral recovery. But I think it shows a good point here that in these two animals that are untreated, we have viral recovery immediately the first time point. Where some of the CRISPR-Cas9 animals that were treated, we have very, notice the log here, very, very small amount of, viral blips. So our conclusions really are that we can design this method in HIV, and we've now have it for SIV to target the LTR and GAG. We've showed cleavage of DNA in vitro, and then brought this into a large animal model showing ex vivo CRISPR-Cas9 editing, and more importantly, in vivo, when we deliver a single dose at 10 to the 13th, we can get viral excision in blood and in lymph node and other tissues like brain and testes. So we also believe that the IPDA shows a decrease in the intact proviral DNA in the lymph nodes after CRISPR, although I will admit, an N of 3 and an N of 1, is certainly just preliminary. And the EDITS assay, although at a single time point, shows the potential that we could really be decreasing the reservoir in that manner. So I just wanted to acknowledge my collaborators. I did that at the beginning, but really myself, Dr. Kalili, Dr. Kaminsky, and Dr. Gordon at Temple University. We were working with our collaborators at Tulane Research National Primate Center, Drs. McLean and Dr. Ling, Gilead and ViiV for the ART drugs for our studies, and John Karn from the Case Western. And also, of course, all of our NIH funding for this study. I do also want to say that we're doing a larger study with 14 animals, or 12 animals, sorry, that is, hopefully we'll have results very soon, and that is from a company called Excision Biotherapeutics, which is different from this study we're doing here, but we're doing a larger funded study for that, and that's going to include, that has multiple doses and multiple time points after infection, so hopefully we'll get to share some of that data sometime soon.
Okay, thank you very much. We're running a little bit behind, but we have time for 2 or 3 questions, quick questions if possible.
Sure, so we're going to hear a lot over the next day or two about delivery mechanisms, a lot about AAV. So to what degree are you confident that AAV9 is the best AAV type, and what's that based on? Yeah,
so really, this is the best delivery system that we thought we have at the time, because we wanted really to do something widespread. So we thought, everything's developing. There's new AAVs all the time. There's new technology. So at the time, we thought AAV9 was the best because it's widespread. And I know the question is going to be, it doesn't go into CD4 cells. We will say we have the same problem in the in vitro system, but in the in vivo, we can see excision, in CD4 positive T cells. So I don't know why it's not, it can do it in vitro, or in vivo, but not in vitro. So. But we're still working on that.
You have a lot of gel-based data, but did you directly quantify how efficient you edited your targeted locus and how many unedited loci remain?
Yeah, so this is something we're currently working on because this is a PCR-based assay. So if you know anything about PCR, you know that it's a qualitative assay. So we are working really hard. We can do that in an in vitro system using GFP, but we're trying, we have a lot of time spent on trying to find really what's the efficiency. And that's something we're working on.
Okay, just one quick question.
Try to be fast.
Given that AAV is going everywhere, have you done a decast chip to look at off-targeting of this CRISPR? Because
yes.
We see about 3,000 off-targets with decast.
So we've done really rigorous off-target analysis, and we're starting with the in vitro. We've done it in the mice, and now we're doing it in the monkeys. So when we designed the gRNAs themselves, we've looked at off-target effects, but we have an extensive panel of off-targets. We can talk more about that offline, but we've definitely considered that for sure.
Okay, great. Thank you very much again.